nutrition, and there is a saying which says that we are what we eat. This concept of we are what we eat, though it looks modern, but if you go back to an ancient way of living, especially the ancient Sanatan way of living, they gave a lot of importance to the food we eat. Not only the food we eat, they actually kind of stressed upon when to eat, how to eat, which foods to eat, which foods to be avoided at what points of time, and probably they understood the idea that a large number of diseases go through their gut. And that's something which we are rediscovering now, where we have understood that a large number of metabolic syndromes, the cardiovascular diseases, the cognitive dysfunctions, cancers can be related to your food. So when you look at sepsis, what exactly do you mean by sepsis? Again, over the last many years, our understanding of sepsis has improved, and we have come to one common understanding that sepsis is nothing but a dysregulated host response, which leads to organ dysfunction, and that is to multi-organ failure. But the bigger question is, what determines your host response? Why do somebody have a dysregulated host response, and why do somebody have a regulated host response? Why do somebody having the same bacterial infection mount a hyperinflammatory response and die? Why do somebody with the same bacterial infection not manifest any symptoms? Well, there are a lot of theories which have been put forward where people suggested that maybe the comorbid conditions have an influence in the way your immune response or your host response happens. For example, people believe that if you are having comorbid conditions like diabetes, hypertension, or your food habits, like if you are an alcoholic, you are a smoker, probably your immune response may be different. So then again, you would see in your day-to-day practice that all diabetics don't die, though they have sepsis. Similarly, all alcoholics don't die, though they have infection. So there should be something else apart from these common factors which may have an impact on the way we respond. And that is where people start to identify and understand that probably it could be the genetic makeup which could be determining the way you are responding to a particular insult. And now you have lots of data which is kind of showing us that maybe if you are made up of specific phenotype, for example, different phenotypes among sepsis have been identified. Similarly, different phenotypes among ARDS patients have been identified. That if you belong or if you have those specific genes, then probably you are going to respond in a different way. But are we talking only about our own genetic material? Because human beings today are no longer considered to be living in isolation. We know that there are millions and millions of bacteria which are living along with us. Be it your gut, be it your lungs, be it your skin. There are millions and millions of bacteria which are coexisting with us. In fact, if you calculate the genetic material of the bacteria or the microbiome which is there with us, it is about 100 times more than our own genetic material. And there is data which also shows that my genetic material may be similar to your genetic material, but my microbiome, about 80 to 90% of the time, will be different from your microbiome. And when you have such a large genetic material which is living within ourselves, well, it is but natural that there could be some influence which would be coming from that microbiome, and if that microbiome is altering, then yes, it could be logically assumed that it can have an impact on the way we would respond. Now, if you look at the gastrointestinal tract alone, there are close to 100 trillion bacteria which are there in the gastrointestinal tract, in the GI tract, right from your oral cavity till down to your anus. And you have different bacteria which are there in different parts of your GI tract, predominantly biofilm and the bacteroid species. But if you calculate the entire amount of bacteria which are there within your GI tract, we have close to about 1 gram of lipopolysaccharide which is always there within your body. 1 gram of lipopolysaccharide is huge. You just need a few nanograms of lipopolysaccharide to kill or to initiate a severe sepsis and septic shock and to kill somebody. Now, despite having such a huge amount of lipopolysaccharide within our system, why are we not getting affected? Because we have definitive, when you are healthy, you have definitive protective mechanisms. And one of the first protective mechanism comes from your intestine. We know that the intestine has 4 distinct layers. You have your mucosa, you have your submucosa, the muscularis, and the serosa. So each layer has a definite role in preventing this bacterial translocation from the gut into the systemic circulation. Your mucosa produces your mucin, which kind of coats your intestines. It also releases your antimicrobial peptides, releases your alkaline phosphatates, which kind of inactivate whenever you have pathogenic bacteria or whenever there are pathological components of bacteria which are released into the gut. Then you have your epithelial cells which are tightly regulated with tight junctions, which make sure that the bacterial components don't come out of the intestinal tracts into the systemic circulation. Then you have the immune barriers. We know that the intestines have a rich supply of lymphocytes, there is a very rich supply of your IgA, and you have the gut microbiome. The bacteria which is there within the body also helps you in protecting against pathogenic bacteria because they produce what is called as bacteriosis, and they help in killing other pathogenic organisms. At the same time, these bacteria compete with your pathogenic organisms for space and food. So you have well-defined mechanisms in the space. Now similarly, if you look at your liver and intestine actually work in very, very close association. In fact, they share a lot of things through your portal circulation, through your systemic circulation, and your bile acids. And whatever materials or whatever happens in the intestines is carried to the liver through the portal circulation. You know that liver has dual blood supply, one from the hepatic artery and one from the portal vein. And liver again is an organ which plays a significant role in your immune mechanisms and protecting you against all these organisms, right? Like for example, you have your macrophages, you have the sinusoidal endothelial cells, your stellate cells, lymphocytes, Kupffer cells, and macrophages within the liver. So whatever metabolites or whatever organisms are released from the intestine through the portal circulation into the liver tries to kind of either inhibit them, modulate them, or kill them. So that is, you have two kinds of a defense system, and that is what happens in a normal individual. But what happens in sepsis? We all know that once you have a oral sepsis or septic shock with a high inflammatory response, then your intestinal permeability is modulated or intestinal permeability is kind of impaired, which leads to dysfunction of your mucus production, dysfunction of your epithelial cells, the tight junctions are impaired, thereby the contents which were much or locally regulated are no longer regulated, and those contents start to fall into the liver at a much higher quantity. Another major thing that happens in patients with sepsis and septic shock is what is called as dysbiosis, which is nothing but the normal gut microbiome, the diversity changes. From what were a normal commensal bacteria, they start to be replaced by bacteria that are pathogenic and which are multi-drug resistant. And if you look at the factors which can influence your gut microbiome, there are multiple factors. Starting from your birth, whether you are born through a vaginal delivery or through a cesarean section, what kind of food you eat, what kind of lifestyle you lead, do you lead a stressful lifestyle? And among all these factors, which can have an influence on the gut microbiome, probably the one factor which has tremendous impact is the use of antibiotics. Now, you may use an antibiotic for an indication which is justified. For example, you have a patient with pneumonia and you use antibiotic for pneumonia, and the antibiotic may work and kill the pathogen in the lung. But at the same time, it also has an unwanted effect on your gut microbiome, whereby it can kill your normal or those bacteria which are kind of commensals within the system. So any exposure to antibiotic, especially if it's broad spectrum antibiotic, then the impact of that on your gut microbiome could be tremendous. So, once the intestinal permeability is breached, then whatever was supposed to, for example, once your intestinal permeability is breached, then your bacterial components like your pathogen associated molecular patterns, your DAMs, and various metabolites, they reach the hepatic circulation. And what was normally happening in the hepatic circulation, where these molecules or bacteria were kind of neutralized, does not happen any further, and they can be released in systemic circulation, which can lead to multi-organ involvement. If you see your liver sinusoidal, basically your hepatic artery and your portal circulation meet within the liver sinusoids, where the liver sinusoids act like anatomical seals in trying to clear up all these muck or the debris. But in patients with severe sepsis and septic shock, even your liver sinusoidal functions are hampered so that this filtering does not happen. Similarly, the volume of your pathogen associated molecular patterns and the DAMs is so significant that the liver with the coexisting liver dysfunction is unable to clear the whole quantity or the whole load of the pathogens. Lastly, if you look at the lipopolysaccharides, which were normally kind of neutralized in healthy individuals, the mechanisms which are in place to neutralize the lipopolysaccharides no longer work. And most of the lipopolysaccharides now start to leak out into the systemic circulation. And there is also evidence which suggests that once these molecules reach the liver and once they start interacting with the macrophages, then it can lead to two types of signaling that can happen. The toll-like receptors can interact with the macrophages, either leading to an M1 phenotype or an M2 phenotype. M1 is a pro-inflammatory phenotype and M2 is an anti-inflammatory phenotype. And in sepsis and septic shock, once the gut permeability is increased, you find that more and more amounts of these bacterial pathogens interact with the macrophages within the liver and within the systemic circulation, leading more towards an M2 phenotype where there is more and more release of inflammatory mediators. Apart from this, there is also data which suggests that this attracts your chemotaxis and attracts your neutrophils and platelets, which we all know are very closely related to the release of inflammatory mediators and lead to organ dysfunction. Now, is there any role for your bile acids? Again, we know that bile acids are key signaling molecules and they are very important in both your lipid and glucose metabolism. They are released from the liver and they go to the intestine. Within the intestine, they undergo certain changes, get converted to secondary bile acids. Now, bile acids also have some bactericidal properties. But in patients with sepsis and septic shock, the composition of the bile as well as the bile acid metabolism can be changed. And there could be excessive deconjugation. Now, once the bile acids are excessively deconjugated, then they lose the bactericidal properties. And they start promoting bacteria which are more pathogenic. So, on one hand, the altered composition of the bile acids lead to liver dysfunction. At the same time, these altered bile acids lead to the changes of the gut microbiota. So, in patients with sepsis and septic shock, this axis between the gut and the liver, in health, it is, it works in a fantastic regulated way where it kind of protects the body. But once that axis is broken in patients with sepsis and septic shock, it starts to be counterproductive and can lead to a dysregulated immune response. Now, do we have any therapeutic options or can we have any interventions which can kind of modify the gut-liver axis? Well, most of these are theoretical. You have the regulating proteins which can, which have been tried where they have tried to look at modulating the tight junctions, but not much of success in clinical practice. People have used a lot of probiotics and prebiotics. Probiotics are live bacteria and prebiotics are basically compounds which don't get digested and then they go to the intestines where they get converted as food for the bacteria. Not much of success again. In fact, there are at least 6 trials where they have looked at the role of probiotics in critical ill patients, and once your intestinal permeability is broken, once your intestine starts to leak, then there is a very, very high probability that if you start using probiotics, they themselves can migrate and can cause bacteria. And that is what has been shown in at least 6 trials. Fecal microbial transplantation again is something which people are trying, where you can try to take the feces. It may sound very gross, but you try to take feces from people who are healthy and then kind of transplant that into critical ill patient. Again, it's more of still in experimental basis, but this is being tried in non-critical ill patients for other indications. And I think in Hyderabad there is somebody who is doing that for autism, mainly to try to change the gut microbiota, because apart from the gut liver action, we also know that the impact of the gut microbiome is there on a lot of other conditions, including your cognitive functions. Exchange resins again have been tried, where they have tried to look at whether you can absorb the various bacterial components. Not much of success. HDL again is something which can neutralize your lipopolysaccharide, but again, as I said, they are more for theoretical purpose as of now. I'm not sure whether they'll be coming into clinical practice very soon. To summarize, well, we have been told repeatedly that sepsis management is all about hit hard and hit early, and we try to do that by giving more and more antibiotics. But please remember that every antibiotic that you give can have a significant impact on your gut microbiome, can lead to significant dysbiosis at the gut, which can have long-term effects. Well, liver, we know that it acts as a gatekeeper between the GIT and the systemic circulation, where it tries to kind of filter out, tries to clear all the pathogenic material which is coming from the gut. But in sepsis, that gatekeeper is breached, and you can have a large amounts of the pathogenic bacterial material which leaks in the systemic circulation, which can have an impact on modulating your immune response and leading to a hyperinflammatory response. Well, as of now, we only have therapeutic options. Nothing seems to be, there available for us to be used as of now in the critically ill patients. Well, thank you so much, and I also take the pleasure of inviting you to the PENSA 2022. This is a major international conference on nutrition, which is happening in Hyderabad. I think you should be attending this because some of the very big names in clinical nutrition, like Vishma, Gail Fishy, Robert Martindale, Vanessa. I mean, some of the very, very big names who have been authors for most of our guidelines are there to present their papers. And there is one session exclusively on the gut microbiome. So I think since this is happening in Hyderabad, I invite all of you to please get registered and attend the conference. Thank you so much.
Thank you, sir. The way you explained the crosstalk between two organs, certainly the intention will be more looking after the lungs, heart, kidney crosstalks. But this is a very nicely explained regarding that. So the wife was a donor, but not very good result because we are very hesitant. It took you 7 days to convince her. So hepatitis was very keen for that. So they did it, but still we are in line. Hopefully.
So just like you talk about the interventions, our gut microbiome is unique to us. As I said, my genome may be similar to your genome, but my microbiome is very much different from your microbiome. So maybe when you have a patient and you are trying to match or do a fecal, you really don't know whether it's going to match with the patient's microbiome. And you also need to be very, very sure that the patient who is donating or the guy who is donating has a fairly good quantity.
So our next talk